who has heard of the term Theranostics before? Oh my goodness. Very informed audience. All right, Louise. This is obviously a very smart group, but for the general public out there, most people may not know what Theranostics stands for. Can you just really take us through what it is and how it works?
Well, for the initiated, it's a mystical religion. No, it's not. So Theranostics, it's a made-up word, so therapy and diagnostics. And it's using these companion, very small proteins, these peptides that have been developed by amazing chemists that bind to receptors on cancer cell surface. And what we can do is we can clip on a radionuclide that is for imaging, for PET imaging, so we can take a picture of the cancer, we inject that into the body, it finds the receptor on the cancer cell surface, it binds to that and gets pulled into the cell so we can see where the cancer is, no matter where it is. And then we can unclip the imaging agent and we can clip on a nuclear warhead. So we can clip on a therapy radionuclide, and that does exactly the same thing. So what you see is what you treat. Is really what Theranostics is. Right. And it's not limited to one cancer type. We can take any receptor on any cancer and use this technology. So we're really starting, and Oliver's done an amazing job developing the evidence in prostate cancer. Mm-hmm. So there is now approval in the US for this, for prostate cancer, and it improves survival.
Amazing. And so you mentioned prostate cancer. Is that the main use of it at the moment?
Yeah, so at the moment there's a receptor on the cancer cell surface called a prostate-specific membrane antigen, and that's a really well-expressed receptor in prostate cancer. It's nice because it tends to get more expressed as the cancer gets more aggressive, so as it spreads in the body, you get more of it.
Right.
So its response in those cells is higher. So what's super nice about that is patients who've got really nasty disease actually can take up a lot of the radiation and treat that.
And are there patients, which are the patients that are most suitable for this type of approach at the moment?
So at the moment, from the work that Oliver's done, it's really, there's a lot of evidence for it in the advanced space, in patients who've already failed chemotherapy, patients who are being considered for chemotherapy who have metastatic disease. But actually the work that Oliver's doing as well is really trying to take that right to the beginning of prostate cancer. And one of his big goals is to avoid what we call castration. So avoid the need for androgen blockade or blocking testosterone in prostate cancer is what you want to do, right? Mm-hmm. Where it will end, we can have endless arguments about whether that will happen or not happen. Maybe it won't happen. But at the moment, we're really at the other end of the spectrum. So advanced disease.
Well, thank you for that sort of brief overview. We'll go to Oliver. How did we get to where we are today? Can you tell us a bit about the history of how it all came about?
You know, you have to go back a long way and understand that radiation can kill cancer. And that goes back over a century. But the idea that we might be able to take the radiation and molecularly target it to a particular cell is much more recent. Initially, we're using bone-targeted therapies. And the bone-targeted therapies, such as strontium and samarium, were able to palliate pain, but they didn't prolong survival. And then along came a certain compound called radium-223, and it's a bone-targeted, not to the tumor itself, but to the stroma around the tumor, and we actually got a prolonged survival. So that was really, really important because it demonstrated you could prolong survival.
So that was the first time that you could actually prolong life?
The first time that we could actually prolong life. Now, the neuroendocrine folks, if they were here, would want to tell you about the important work that they've done treating neuroendocrine cancers, but it's a relatively small cancer compared to prostate, which is big. And then the more recent one is a PSMA 617 lutetium, which in advanced disease after chemotherapy and hormonal therapies had failed, we were able to prolong survival, which is a very important metric. Now, being able to bring this earlier is one of our goals, as Louise was mentioning. Eventually, we'd like to be able to get rid of hormonal therapy and chemotherapy because men don't want to be castrated and they don't want to get chemotherapy. So we have a lot of ambitions about how to do it. People like Alan are going to enable us to take the next step.
All right, let's go to Alan then. So how do you fit into this amazing work, Alan?
Firstly, I work with these amazing people, have this amazing team over here, and what we do. So I'm a scientist from this precinct. Came out of the Garvan. It's great to be back here. And scientists work a little bit different. Scientists go, this is what we have today, how do we make it better? And so we went back to the drawing board on some of these agents that we're speaking about right now, and worked very closely with the scientists that we collaborate with and said, here's the current standard of care. This is what we're looking at now. This is the one that Oliver worked on in late stage. We said, how do we build a molecule which is better? And how do we make it better, not just for therapy, but something we didn't touch on was that the focus is on therapy at the moment, but with better diagnostics, we actually have potential for better treatments. And that can be, in prostate cancer, can be earlier or looking at detection of cancers outside of the prostate when you're making decisions like whether you want to lose your prostate or not, when prostatectomy, but then also in biochemical recurrence. So that next stage when you've had that, when the cancer comes back, if it comes back, we can detect it very early. And Louise and us are doing a study now looking to get very, very early in that disease state, and maybe treating with external beam is the therapy option, but you can't find it necessarily without these great diagnostics. So we went about building much better diagnostics. And every day, because you asked a great question, we're constantly looking at what's next, what's next. So yes, neuroendocrine tumours was there. Some of the group that actually worked through the translation of radium chloride are in our team.
Amazing.
Yeah, the CEO is on our board, and a few of the team is actually part of our organisation, because that was the first step. That was passive targeting. That was passive uptake of a salt into bone mets. Now we're looking at active targeting. How do we build new molecules that actually physically attach to the tumours, irradiate the tumours, without hitting too much off-site targeting for side effects, reduce side effects and excretion. So we published a paper only a day ago, which was in preclinical, which is in breast cancer. We're looking at breast cancer with hitting HER2, which is well understood.
We've done a trial in breast cancer.
We've done a trial in breast cancer with another agent we have as part of the portfolio. So we're excited. But we think it's very new. So there's so much opportunity. People think we've answered all the questions. We haven't yet. And we're getting better and better at treating these cancers. One
thing we do is this academic industry kind of partnership. They've got the product, right? But I can come and say, look, I want this kind of trial, because that's not something you're going to do, but it's something that will really influence the way we treat our patients. So this kind of academic industry partnership is incredibly useful at answering questions that wouldn't otherwise get answered.
And treat people in Sydney.
Yeah, so
I love this city of ours.
Exactly. I was going to ask Louise, Louise, take us through the patient experience. How does a patient access this treatment? You know, particularly what's the criteria, and how many patients are you treating at the moment using this technique?
So we do have a clinical program at St Vincent's, but it's really difficult treating in Australia at the moment compared to Oliver. Oliver has funded treatment in Australia. In Theranostics in Australia, there are no federally funded treatments at this stage. So we have evidence to show it improves overall survival. We know that it improves quality of life. People live longer, but we don't have funded treatment. So
can I ask why? Is that because it's been knocked back for approval, or it hasn't been put forward for approval to be funded?
It's been put forward for approval multiple times, but the problem in Australia is we have very high bars for funding. From a federal government point of view, you have to prove overall survival to a very high level and quality of life years. So it's a higher bar than the bar that you have to have in the US. So great approvals in the US, approved in Europe, but not yet approved in Australia. So patients can access it at St Vincent's, and we still have a clinical program. But what we try and do is we try and open as many trials as we can, so we can give funded treatment to patients here. And so we put up our hand continuously to open trials. We have a fantastic relationship with oncology, and Anthony Joshua is going to be here tonight, but isn't. But we work very, very closely together to open as many trials as we can to get our patients access in Australia.
And I'll just ask Alan and then all of you. So this sounds like an amazing sort of mix of chemistry and biology and physics and a number of different branches of science coming together. Why has it happened like that we've got this sort of synergy of so many different things coming together to create this theranostic approach? Alan and then Olivia?
Yeah. So I'll start by, it is great chemistry, great biology, great physics. And that's why I find it really amazing. And when I first came across it over a decade ago, I thought it was incredible what we can do. And it was something that I understood with a background in biochemistry and molecular biology for a change. So, but the drive to it, so you need this, the understanding of the biology, the receptor interaction. We do use physics in the use of radiation. So we use a clarity, a combination of 2 isotopes of the element copper, and copper-64 for imaging and copper-67 for therapy, which are terrific. We had no doing in the physical characteristics of that. Whatever you believe in, God or nature or whatever, produce those isotopes, and they're amazing for the age that we live in now in this radiopharmaceutical space. And now we're just constantly using chemistry and biology to understand how we can target these new cancers. And so it tests every part of your scientific acumen to try and understand how you produce new molecules. But that's what makes it exciting. And we're now escaping, if you think about cancer care was really the 3 pillars. Surgery, cut it out. External beam radiation was zapping it from the outside in, and that hits everything on the way in and through.
You've got an oncologist here, though, so
which can answer that, and then obviously chemotherapy. So this next step, this active targeting of thing, is really years and years of antibody development first, and then understanding of how peptides work, and now we've been able to capture all that radiation as well as the sort of biology components to really now grow this area.
Yeah, and Oliver, so what drew you to this branch of all the branches of oncology and medicine? What made you interested in theranostics?
The potential and the effects on people's lives, and the lack of side effects that most of these agents actually have. So, I was raised in the chemotherapy era, and we kind of had the full dose on time mentality, and we did very punishing chemotherapy in hope to eradicate tumors. And some we did, like testicular and a few of the lymphomas, a few of the leukemias, we could actually make a lot of progress. But for the solid tumors as a whole, even though the progress was tangible, it was not good enough. And when you begin to understand the power of theranostics and the physics and the chemistry and the biology all put together, I think we have a really high ceiling. I think we can take it further than the chemotherapy has gone. And that really is the promise and the reason why I'm here and why I do what I do.
And Louise, what about you? Like, of all the branches of med, I'm always fascinated with doctors and scientists to ask them, like, because the world is their oyster. If you think about all the different branches they could have gone into, what drew you to this and to make this your thing that gets you out of bed in the morning?
I mean, I think it was a fairly torturous journey for me, but I have to say that what keeps me here is the science. I love the fact that you're dealing in copper. I use lutetium, I use actinium. There's a company sitting just there that's using lead-212, so Advanced Cell is here tonight as well. And they're doing great work as an Australian biotech in another element. And then we also use technetium, lutetium, actinium. It's massive in terms of what we can use, and it's fascinating.
And I mean, just maybe also, can we explain to the audience as well, like the difference between, say, the approach with chemotherapy, where chemotherapy kills off any fast-growing cell in the body, where this is so targeted to and so precise to just attack the cancer cell and very little else around that?
Well, I mean, chemotherapy diffuses everywhere, and virtually any rapidly proliferating cell will have an effect by the chemotherapy. And so people's hair fall out. Sorry, Alan, you're not going to say much more, but these things could happen. But I mean, you have effects on the bone marrow, you have effects on the hair follicles, the GI tract, and more. Whereas this sort of precisely targeted radiation is able to go to the tumor selectively, and of course that's our job, is to find the perfect match between the isotope of the target of the cancer cell to be able to have a therapy with virtually no side effects. But we are having side effects. I don't mean to diminish the fact that we do have side effects, but we're much, much, much better than chemotherapy. In fact, as Louise has helped to lead a trial that compared the side effects of chemotherapy and the lutetium, and without a doubt, the lutetium patients had less side effects and better quality of life.
And just staying with you, Oliver, so you're part of here as part of this amazing program of exchange with St Vincent's clinical exchange program. and you're a world leader, as we've heard, with your expertise. Why did you take up the opportunity to come? We're very glad you did, but why did you take up the opportunity to come to Australia?
You know, there are a couple of reasons, and Louise Emmett is one of them. She's a unicorn. She's an absolute unicorn. I don't know anybody in the world who's quite like Louise, and she has done amazing work, and I wanted to come and learn from Louise and what's going on at St. Vincent's, and that was a very significant driver. But there's also the biotech, represented in part by Alan, but also the mention of advanced cell a little bit earlier. There's another company called Telix that is there. And these three Australian companies are actually doing beautiful, beautiful, beautiful work. And they're world leaders, international leaders in the theranostic space. And I believe, and I've been able to meet with leaders within each of these three companies. We'll be having dinner with Alan tonight. And that industry academic partnership alluded to by Louise a little bit earlier is really key. And it turns out in Australia it's going super, super, super well. There are also regulatory factors, I think, that feed in. And now you have access to capital that's making a difference. So between the Theranostics that are in part led by Louise, the unicorn, and by Alan, who's up here on the panel, and Advanced Cell, who's not on the panel but represents a really important advance as well, there's a certain milieu and activity here that is intoxicating. It's really, really good. And by the way, thank you to the American Australia organization for funding and to St. Vincent's for the opportunity.
Oh, we're very glad to have you here. And Alan, so you've heard how excited Oliver is about what's happening in Australia. Just tell us a little bit more about the role Australia is playing in this space, in theranostics. It sounds like we are really, kicking well above our weight when it comes to what we're doing in this area.
Yeah, it's a tricky one. So I've been passionate about this translation of science for a long time, and I don't want to say that we do it well, because I don't think we do, and there's a number of reasons why we don't. But it's great to see Advanced Cell, for instance, Aussie, homegrown as a play, and Clarity is 100% homegrown. And I think when we say Australian companies, we have to be cognizant of that. I worked in the capital markets for a long time, and I worked with a lot of US companies. We listed them here in Australia, and we would never view them as Australian companies, and even though they're listed here. I'm a bit more of what are the genetics? And I don't think we have enough companies which have the genetics that are Australian. Clarity is a little bit different. Clarity is 100% homegrown from the benchtop. Good science. It was from Melbourne, but University of Melbourne. But with a great scientist down there, Professor Paul Donnelly, who we constantly have a collaboration with, and translation has been on original molecules around the cage that holds the copper isotopes through to new molecules that we've developed. This is what drives me. And Oliver, obviously, is allergic to maybe Australian translation. But very focused on that we have to be cognizant that when we say Australian, it is about driving this science translation. And there's not enough. I think we should do better. But the reason why it worked at Clarity is because we had outstanding basic science. We had a fantastic basic scientist in Paul. I want to date back, actually. The original work was done at ANU some 70, 80 years ago, because this never happens overnight. There was a guy named Professor Alan Sargerson. He was a chemist who said, we hold these other metals with these other cages, but we can't hold copper or cobalt. So I'm going to make a cage which holds copper. And then that was great, got into the hands of Paul Donnelly. He said, well, I'm going to make it better. I'm going to make it functional. And importantly, bifunctional. Not only does it hold the metal, but then we can attach everything to it, like an antibody down to a small molecule, which actually targets the cancer. And that was great initial work. We translated that straight away into the clinic in Australia. Then we worked very closely with Louise along that path in phase 1, phase 2 trials, and built up a cache of intellectual property. Now we have 30 plus patent families. This might, Bill might, also with Advanced Cell understand this process. IP is incredibly critical when you're trying to raise capital. The ASX is there. We've been on the ASX now for 3 years. We just moved into the ASX 200. I think it's all here, and I think we should do more of it, given our expertise, particularly in this theranostics nuclear medicine space.
Louise, did you have anything to talk about in terms of why Australia is so well placed to be a leader in this area?
Look, I think we're well placed and we're not well placed. There are difficulties here in what we do. We have trouble getting funding for our patients. I want to see my patients get funded treatment, and in the meantime, I'll open trials. I'll continuously open trials to get access to our patients to fantastic treatments that really help them. We've got a lot of work to do in Australia with that. I wouldn't say it's all rosy.
And
I
Oliver?
want to comment a little bit. I think both of these are a little bit modest. I think the Australians are leading the world, and there are other legitimate hopes out there, some in Germany, Switzerland, and if you look at the big boys, then probably the Swiss are number one. But in terms of innovation, it's remarkable what's going on right here.
Sounds good. So, Alan, you mentioned your product uses copper. Can you explain a bit more about that for the audience who may not understand why copper, and what are the advantages and what are the benefits that we might see with that product?
Yeah, so why don't we stick on one product, the prostate cancer product that we're working very closely with Louise on. It's called SAR-BIS PSMA. It's called SAR because
That's a catchy title. Woo-hoo.
Yeah. It's SAR because of the cage was called a sarcophagene by the scientists some 70 years ago. Sarcophagene after sarcophagus. What goes in doesn't come out. They're so innovative, these chemists, BIS means that we build a bispecific. We actually have two targets that target the cancer better. So we increase the amount of uptake and retention over time into the tumour. And then the copper components fits in the cage, and those dynamics, the physical characteristics of copper 64 and 67 are ideally suited, as I mentioned. But copper 64, the benefit there is the current agents that we're talking about in prostate cancer for diagnosis actually have a 1 hour half-life. And you don't have to be in a 2 hour half-life. So you don't have to be a scientist.
Just for the audience, explain what that means.
Actually, I might explain
Why don't you
it.
do
How about
that?
I explain it?
You explain it.
Yeah, I'll explain it. So what I do is I look at prostate cancer every day. I do images, I report images in patients every day. So what happens is when you look at a prostate cancer, if it's only got a few receptors on the cancer, sometimes it's not very bright, you don't see all the cells. What Clarity have developed, instead of having one receptor binding to one peptide, they've got two peptides that are like that bind to the cancer cell, and instead of seeing twice as much, you actually see 3 times as much. So when we look at the image, instead of me having to squint and put my glasses on, it's very, very bright.
So you're getting a much better quality.
Yeah, so what we have to do is we have to
on.
innovate in terms of, you've got the receptor on the cancer, but what can change is what we bind to it. So we need things that bind much better, that are much brighter, that make it much easier for me to see. So I can walk to the end of the room and I can say exactly what's going on. It's much easier, which means we don't get it wrong, we get it right. And then when we clip it onto a therapy agent, we get a much higher dose into that radiation dose.
Where it's really needed.
So it's transforming how we can look at the images and what we can see.
Amazing.
Can I finish off
yeah,
on
yeah,
our
go
product
on.
as well? Is that
Okay.
Go on.
Okay. The second part to that, which the first part was amazing, wasn't it? Right. So you're getting much more uptake, much more retention. You can see, but copper 64, you're about to remind everybody, has a 12.7 hour half-life.
So that
Not
means
a 1 hour
that's how long it stays in the body, doesn't it?
No, that just means it
Half-life.
loses half of its energy over that period
I
of
was
time.
taking that into account.
Right.
So what happens is you can not just image at 1 hour.
Right.
You can image it 4 hours, 6 hours, 24 hours, 48 hours. And when you image later, you can imagine this. You don't have to be a scientist to know this. It basically, when you infuse it, it's swimming around the bloodstream. It finds the receptor, it binds to the receptor. The longer it swims around, the more it binds to the actual tumour. And then what happens, the background washes out over time. So you have what's called, you can call it many things, increased signal to noise ratio, increased tumour to background ratio, or increased contrast.
Or easier to see.
So basically, it becomes easier to see for the oncologist and the doctor.
And at 24 hours, from ours at that 1 hour, which is already better, there's actually a 5 times increase in that signal to noise ratio.
But what we have to do is academic trials that actually prove that. So we can say we're fantastic and it works really well, but I want to see an outcome in a patient. Actually, I want to see how it changes for a patient.
Yeah. And you would hope, you would logically thinking
Are you
that...
trying to argue with me on
No,
that?
no,
Okay.
I was told this
I've got
might
a next
happen.
one. So what we did was, we did a trial with Louise.
Oh, we haven't done it yet. We haven't done
We
it
did
yet.
Propeller. We
Oh,
did
Propeller, yes,
Propeller.
we did Propeller.
And we did Cobra. So we did these, and we did.
Yeah.
So we try and do as much as we can in Australia with Louise, but a lot of the stuff, and this is a little bit of an issue again. The centre of the world is where Oliver is in our space in the US, because they do pay for therapies. In Australia, we don't like paying for therapies. We have a small population, is the first issue. The second part, we don't like paying. So when big pharma comes here and goes, would you like our products and are you going to pay for them? Australia goes no, and they say, well, no one lives here anyway. We'll just go somewhere else. We did that during COVID with the Pfizer vaccine. We ended up with AstraZeneca. Right? So Australia doesn't have much buying power. So we try and run as much here with Louise, but predominantly with Louise because it's great. She's great and it's a great facility. We did Propeller here. Which was great in pre-prostatectomy.
We haven't done the real trial though.
And now we're doing...
Haven't done the real trial, which is the head-on comparison to see
So we're
how
doing
good you really are.
So typically,
right?
typically a pharma company would not do a head-on comparison.
No.
But we're so confident in our product, and there's probably going to be a few questions this later, that we're keen to go head-to-head. Now...
So just explain, so that trial, Louise, that you would be wanting would be basically to say, let's look at this copper way of diagnosing prostate cancer versus, say, what you're currently using.
Yeah, so we have standard
and
of care.
basically see how much better it is, how much more accurate, and whether it leads to better outcomes for the patient.
So how does it change the management of that? So you can have something that's brighter, but is it actually better for the patient? So when I look at better, I look at, does the patient live longer? Does the patient have a better outcome? Do they have less toxicity? Do we pick up more for that patient? Is it better for the patient? And so we have lots of metrics that we use. So by a head-on comparison study, it's really nice to try and figure out whether it really is much better or not, or whether it looks better but doesn't actually change anything.
Yeah. And so I just want to talk about, Louise, a little bit more about what's happening at St Vincent's and about some of the research that's being developed, things like validating biomarkers and things like that you're doing.
Yeah, so we do a lot, particularly in the prostate cancer space, and we are busy trying to figure out when we do our imaging, how should we report imaging? All of these things are very new. You can't just say there's a hotspot here, there's a hotspot there. We need to know what it actually means. What is treatment response? What criteria should we have? So we do a lot of trials both in imaging and also in therapy. Not only do we have this fantastic therapy agent, which is targeted, but what combination of treatment should we combine it with to actually get better outcomes? This is something that Oliver does a lot. And it's about making people live longer, better lives by using the agents that we have in a very good way to get much better outcomes for our patients. And you have to do that in a clever way, don't you? I think you can really answer that.
Oh, well, I think you said it really well, and I'm not sure that I would add that much, but as we begin to add in radiation, there are also other components of cancer therapy, whether it be external beam, whether potentially chemotherapy, radiation sensitizers, DNA repair inhibitors, and more. And we probably will be assembling the building blocks to be able to put things together with the idea that one day we're going to cure people. And right now we're in more of a palliative mood, but I really can see the possibility of cure if we get the combinations and doses and schedules right. And that, keeps me waking up in the morning and really happy to go to work.
And so what are you seeing over there in the US with Theranostics that we're not quite there yet in Australia? Like what could we look forward to that you may be already having the benefits of there that we haven't quite reached that point yet?
I think you're actually the leaders. We're the followers. Now, it turns out in the original dosing of the trial that Louise alluded to, which showed a prolonged survival, we actually picked up the dosing from Australia in order to put the trial into place. Now, one of the things that big global companies can do and the US can do, we have deep capital markets, we have deep investments, and a lot of people don't realize how expensive these things are. Like the big trials that I'm kind of running right now, $400 million, $500 million to do the trial. I mean, it's real money. And it takes a global series of partnerships. Now, we'll say that the American capital and the European capital, the global capital can get ahead. So the capital markets are better elsewhere than they are in Australia, but I think the innovation is really leading here.
And Louise, I might just get you to talk to the audience about the role that Philanthropy has played in developing Theranostics at St Vincent's. Can you talk a little bit about that?
Yeah, I can. So next week I'm presenting a randomised trial that we did around Australia, which has shown very significantly improved overall survival in men with metastatic prostate cancer. I think you've been saying it's the best improvement in overall survival.
Why don't you let me say it?
Yeah, awesome.
Because
Yeah.
she is going to present the best survival ever reported in these metastatic, what we call castrate-resistant patients, ever reported.
Wow.
The single biggest effect ever reported. Right here. Led by her, conceived by her, and funded in part through Philanthropy.
It was. So, in fact, this was a $5 million grant that we got through the Prostate Cancer Research Alliance. It's in Vincent. And I was missing, the kick in the tail of that grant was you had to have $1 million of institutional co-funding to be able to put the grant in. And I was $300,000 short.
Wow.
And that came from a single donor who was the friend of the friend of a friend of a patient who I knew, and I will be forever grateful to that patient.
What amazing impact.
And he's so pleased with the outcome, but it's going to change the way we give the therapy. It's going to change our combinations and how we improve patients' lives. And that's super, super exciting. I think that with research and funding research and supporting research, you can change the lives of millions.
The flow-on effects are huge.
Yeah.
And can I share from our perspective? So that gives us now, this research, this insight gives us opportunity now to plan what would be our phase 3 trial, to utilise that information with Louise and Oliver. Louise in particular, and how that was structured.
Oliver.
And both. They're both on our scientific advisory board, see? So it's to structure what then would be the best outcome then for patients, we hope again. So we're constantly making these iterations as our science works. And but obviously, Louise being able to do her trial and that combination therapy and being able to do it here just gives us real insight now as to understand how we run our trials.
And that donation came 48 hours before the grant was due.
You must. So what was your reaction when you found out about that grant?
Exhaustion.
Just in the nick of time. How amazing. So we've heard about some of the amazing things that are going on, but there are probably some challenges still, it sounds like, as well. And I might ask you, particularly if we want to expand the theranostic program at St Vincent's, Louise, like what's your wish list? Things like the actual way you develop and deliver the treatment here. Do you have a dedicated space to do that, or?
No, not at the moment. We don't. It's fairly rudimentary, our space. And if I was going to choose between rudimentary and funding trials that actually change the way we treat our patients and getting access to our patients, I would 100% of the time choose access and trials because they're our future. And they give our current patients things that they just can't access otherwise. So I would say trials, research. Research takes a lot of staffing. It takes, to do really high quality research takes people who are dedicated to what they do at a trial level. That's really my focus.
And let's just project a little bit in the future before I turn over to the audience to ask some questions. I'm going to ask each of the panellists where they see this branch of medicine. Let's project 10 years into the future. What are your hopes? What would you like to see happen with this amazing branch of medicine? Oliver, we'll start with you.
Well, we've begun to talk about targets and a lot about prostate cancer, but we heard the mention of breast, but I can imagine that the diffusion of this broad technology will occur across many, many, many cancers, that it won't just be prostate and neuroendocrine, it'll be breast, it'll be colon, it'll be brain, it'll be lung and melanoma and more. So diffusing across multiple diseases. Right now we have a couple of different isotopes, and the copper 6467, perfect pair, the lead-212, very provocative, but I think we'll have even more isotopes to be able to push forward. And we'll talk briefly about combinations, because I think that combinations can be very, very, very productive, being able to hit the cancer cell from multiple different perspectives and have non-overlapping toxicities, and those combinations are going to be key. So more targets, more cancers, more isotopes, and more combinations. And it's going to happen quickly, very quickly. 10 years is not that long.
Wow. And Louise, what's your future? Like project into 10 years into the future, if we come back into this room, what will you be hoping that we'll be talking about when it comes to theranostics?
So we'll be using molecular imaging really well, but we'll be using it with AI that not only tells us how many spots there are, but we'll have deep insight into the phenotype. We'll be able to look at heterogeneity. We'll combine that with genetics. So someone will come in, they'll get a blood test, they'll get their genetics done, they'll have their quantitative imaging, which will be AI driven. We'll be able to tell them exactly what treatment that they need at exactly what space and with what combination, because Oliver's completely correct. It's not just this technology, but it's the combinations of the technology that are really going to drive survival. So I think AI, and as you said, just all the different isotopes, lots of different peptides and lots of different new tumour types.
Amazing. Okay, and what about for you, Alan?
So it's interesting because scientists have to do it. So we're in the midst looking at what that future looks like. And I must say at the moment, and we had a conversation earlier, Oliver, a little bit disappointed in where innovation is. I don't know another radiopharm company that's necessarily built their own product, for instance. Usually it's bought from here or here. A lot of the products which have been transacted upon by Big Pharma are generics. There's been 3 or 4 transactions on that. And there was a little bit of a misstep by J&J who thought they'd build the biggest antibody with the biggest isotope in actinium and started killing people, not realising that that's not a good thing. So I think right now we have to get set, and what we say, it's not about, we try and find this concept first, and I know everyone's going out on different isotopes. We focus on a thing called the Goldilocks zone. Not too hot, not too cold, just right, where you can treat the cancer, but not have the side effects. And that is critical, and we've seen at the moment side effects that I would view as unacceptable. So we're very tight on that, looking at dose-limiting toxicity and things like that in our programs. And copper-67 is ideally suited to that. It's a beta, much like the PSMA-617 that Louise was talking about, Pluvicto, which is the Novartis product. I think strategies in that of making alphas a lot safer is going to be critical.
When you say alphas, can you explain that to the audience?
Just in simple, betas are like softer. They're nicer. They go and kill the cancer. But when they hit off-site targeting, they're not as destructive. So we see even when we've got off-site targeting, we don't necessarily lose the function of the tissue.
And when you say off-site, you mean so when it hits something other than the
healthy
tumour?
Tissues.
Yeah.
It's healthy tissues, then we don't hurt the healthy tissues. With the alphas, and we hope that obviously Bill is in an alpha with lead-212, which has a much shorter half-life, so hopefully there's benefits of that. The alphas hit with a bang, but what it hits, it kills. So
I wish it did actually.
Well, I wish it didn't sometimes. I wish it didn't kill everything it hits. And so that means we need much more precise targeting moieties, much more precise to hit the lesions. And so we have to work on that. So when we're working with alphas and betas, combination, the betas will have a strategy, the alphas have a strategy. The betas will go much earlier stage. We'll try and treat cancers a lot earlier, given the safety profile that we have in those. So that's what we're looking at, get much earlier. And we want cures. And whether it's a diagnostic strategy that we're working with Louise on, imaging much smaller cancers and then treating them with whatever we can, that's a potential cure, getting it early. And then when we work on these new molecules, we're looking at antibodies with pre-targeting. This is really exciting if I discuss that for a second. Pre-targeting is when we have the antibody and the target there, it goes in, finds the cancer. We image a couple of days later and then we treat. If there's lots of it on the lesion, we treat with the therapy 4 or 5 days later and kill the cancer specifically. We've got great preclinical data doing that. We've worked with IO. We haven't discussed much about IO today. We've got some great preclinical data where immuno-oncology, you've probably heard of drugs like Keytruda. Keytruda actually doesn't work on a lot of cancers. So
do people know what immunotherapy is when it comes to cancer treatment?
Yep. So, because I can describe it if you like, but
maybe just give a look. Just
what it is from a scientific perspective is there's a protein that makes cancers invisible. And so what this does is hit that protein and then make the cancers visible to the immune system. So then the immune system kills the cancer. Usually has about a 20-30% efficacy rate. What we've found in Radiopharm is that we can actually turn some of those cancers from off, not getting affected,
working,
to on. So at least additive and potentially synergistic effects. We'd love to see that in humans. But this is why I'm a scientist, you're a clinician.
Yeah, I know. I know. So I was going to say, I'm not sure a human being wants all of that because maybe just the tumour itself. It's a shame we can't isolate the tumour, give it the immunotherapy and avoid the side effects. That's what you're talking about. We've been sitting on a beach talking about all of this for hours and hours and hours.
Yeah, but if we don't do it, we don't know. So, and there's not a lot of work in this space yet, and we would love to see it. So we could have given up years ago and said we're never going to do this.
So what do you think, Oliver?
We're going to do it.
Exactly.
I
think IO is a very exciting prospect, and so do a few.
Well, we've been doing an IO trial with lutetium, and we just shut it down because we had a couple of deaths.
Because there's prostate as well.
So you do need to be obviously very careful.
There are many different forms of immunotherapy, and looking at the current, what I call the PD-1s and CTLA-4s, is one particular approach, but immunotherapy is evolving rapidly too.
Yeah.
And there's some beautiful personalized vaccines that are coming out from some of the RNA companies that are quite provocative.
So this is a vaccine for once you have cancer. It's not a vaccine to prevent something, isn't it?
It is. Yes. It's designed to be able to treat the cancer by using a bespoke process to identify the proteins on the surface of the cell that can then be upregulated in a very specific way, driving the immune system to that cancer. So it's some pretty provocative stuff.
In a sense, Louise? What it is, you confuse the cancer. You attack it from so many different ways, in so many different ways, that it just kind of goes, oh my God, and gives up.
It's all too hard. I'm out of here. Sounds good.
So if we just touch more a little bit on the science then, because the science part, we can make new cages. We can make new cages, the whole isotopes. And that's something we're doing right now.
Very important.
And looking at new strategies with different isotopes. We think the science is amazing. We think we're only just scratching the surface. So to say, no, we can't do this, no, not for us. We want to give it a go. We want to develop like we do. We make new products.
I meant IO, right? Just the IO.
I don't care what it is. We're going to try everything, and we can do it in first, obviously, preclinically, and then look to move it to the humans when we think it's safe. And that's always a strategy.
Well, Alan says it very clearly. We're at the infancy of this field. We are really just beginning, and I see an absolute explosion of possibilities over the next decade and even longer. Again, with these targets, this combination in different diseases and different isotopes, and the way we're going to combine therapies in the future. And it's going to be an incredibly vigorous field for more than one decade, probably 2 to 3 next decades. It's going to be phenomenal.
Amazing.